Thank you very much, Yechiel. So we'll move to the presentation. So nature's gift, biomaterials for additive manufacturing. Before I start, I'd like really to quote the former Minister of Oil of Saudi Arabia, who said that the Stone Age did not end for lack of stone. And the oil age will end long before the world runs out of oil. And I think for a very good reason, because biomaterials are so much better. But we shouldn't criticize our scientists for not making really good synthetic materials for a simple reason. We didn't have enough time. 200 years of modern chemistry is not enough time to make really good materials. While nature had 3 billion years to develop some of the most amazing materials that we wish we had in our possession. Yet our orthopedic surgeons continue to screw and glue synthetic materials into our body, and I don't think that this is a great idea. Why? Because they fail. Many times they fail just like this plastic fork because they're not strong enough for the performance, but other times they fail for other reasons, and mainly because their biomechanical properties do not really fit their surrounding tissues. But the real reason why it's not a good idea to introduce synthetic material into our body is much more fundamental. It's because in nature, things are done differently. In nature, nobody takes my head and screw it into my neck or take my skin and glue it onto my body. In nature, everything is self-assembled. Every living organism, whether it's a plant or insect or fish or human being, all made of cells, and the cells contain DNA, and the DNA encodes 4 nanobio building blocks. Many times there are proteins, other times there are enzymes that make other things like fatty acids, polysaccharides. But the common feature of all these nanobio building blocks is that they don't really need us. They recognize each other and self-assemble into scaffolds on which cells are growing and proliferating to organize into tissues, and the tissues organize into organs, and together that's the organism or life. So we, about 16 years ago, decided to focus on probably the most important nanobiomaterial for us, for human being, which is collagen. Why collagen? Because it accounts for about 30% of our dry weight. We have nothing in our body more than collagen except for water. So I always like to say that anyone who is in the business of replacement parts for human being would like to have collagen. But admittedly, before we even started our research, they were already in the clinic. More than 1,000 medical implants, all made of collagen. You know, simple things like dermal fillers, artificial meniscus, bone grafts, and even leaflets for heart valves. So all made of collagen. Well, then you should ask me, why did you bother? Well, the reason was that the source of all that collagen up until recently was dead bodies, dead pigs, dead cows, and human cadaver. So that's not a very safe source. And in fact, the FDA already in 2007 made a notice asking companies to start and look for better and safer alternatives. So that's exactly what we did. We cloned all the 5 human genes from humans that are responsible to make type 1 human collagen, and by genetic engineering, introduced all the 5 human genes into a single tobacco plant. So now the plant can make human collagen. The technology was matured, and today in Israel, we grow about 20,000 square meters of greenhouses all over the country, where the farmers receive these small tobacco plants that looks like regular tobacco, except they contain 5 human genes that teach the plant how to make collagen in its leaves. We grow them for about 50 to 60 days. We harvest the leaves, and the leaves are then transported by cooling tracks to the factory where we start the process. So if you know how to make pesto, essentially you know how to make collagen. You just crash it with a very big food processor like machine. We get the juice, concentrate the protein. Protein is then transferred to clean rooms. At that point, we purify the collagen, and the end result is collagen, which is identical to what we have in our body, except it's much safer. Why? Because plants do not harbor human pathogens. And from that collagen, we make different medical implants. So plant-derived collagen is by far much better than tissue-extracted collagen. On the right-hand side, you can see a picture of animal-extracted collagen, and you can see that the triple helix is deformed. So the three-dimensional structure is not kept along the chain. That, unlike the plant-derived collagen, which has perfect collagen three-dimensional structure, keep its structure, and therefore the integrin binding site that enable the cells to bind and proliferate is much, much better in the plant-derived collagen compared to the animal-extracted collagen. And that results in better biofunctionality, superior homogeneity, and improved safety and greater purity. All that is not just claimed, but actually proven by peer-reviewed scientific papers that we published in the field. We can see here now is a comparison of tissue-extracted collagen from different sources, commercial sources, and the plant-derived collagen in promoting different cells that are responsible for skin regeneration. So you can see a dermal fibroblast, endothelial cells, and keratinocytes. In all cases, plant-derived human recombinant collagen surpass the other scaffolds in its proliferation. Second is biocompatibility and non-immunogenicity. And as you can see here on the left-hand side, positive T cell proliferation response, bovine collagen responded, while human recombinant collagen showed absolutely no response, indicating non-immunogenicity. On the right-hand side, you can see also the result of activation of macrophages and secretion of IL-1 beta, which is a pro-inflammatory cytokine. You can see again the level secreted by contacting these cells with the human recombinant collagen was significantly lower compared to the bovine collagen. So we wanted to take it a one step further and see if we can really make strong materials from that human recombinant collagen. So we took advantage of a common technology, which is called wet spinning, that is already a very common technology in the textile industry. And by wet spinning technology, we've been able to generate extremely strong and aligned collagen fibers. These fibers are so strong, in fact, they're 6 times stronger than human Achilles tendon, which is quite remarkable. Another interesting biomaterial that we worked in the last 4 years is keratin. Keratin makes the outer layer of our skin and makes it very important to protect our body. In addition to keratin, melanin is very important to protect our body from UV light. In fact, in our body, we have 2 kinds of melanin, eomelanin and pheomelanin, which are responsible for the different shades of the color of our skin. So we've been able to extract keratin and to synthesize eomelanin and pheomelanin, and from that we've been able to develop high-resolution keratin, eomelanin, pheomelanin bio ink for printing of second skin. So you can see on the right-hand side, a high-resolution picture of face of a girl that was printed by high resolution with that very interesting new bio ink. On the left-hand side, you can see the 3D printing of the keratin and a successful growth of, in this case, fibroblasts that intimately bind to the keratin and grow along the scaffold. So every kingdom actually gave us some wonderful materials. Take, for example, the insect kingdom. Cat fleas are amazing because they can jump as high as 200 times their height. And this is quite amazing because it's the equivalent of a person standing in the middle of Manhattan and in a single jump go to the top of the Empire State Building. Now, you should ask yourself, how cat fleas do it? So it turns out that they make this wonderful biomaterial, which is called resin. Resin is a protein that in simple words is the best rubber on earth. You can stretch it, you can squish it, and it doesn't lose almost any energy to the environment. So everybody would like to have resin, but the question is, how can you get it? Well, to collect cat fleas is not a good idea because they're jumpy and small. But that's the beauty of technology. By simply catching one cat flea, extracting its DNA, one can clone the resin gene and introduce it into a less jumpy organism, like plant or bacteria. And that's exactly what we did. So now we have the ability to produce a lot of resin. So what can you do with that? Well, a lot of things. Here, I just prepared an interesting video showing the effect of resin when introduced into a cellulose sponge. So I'd like to show you now a short video how it can be done. So you can see on the right-hand side a cellulose sponge that is actually without the resin, and you can see only plastic behavior. On the left-hand side, it is exactly the same sponge, however, now together with resin. And you can see clearly that it's extremely resilient. You can push it again and again and again, and it always goes back to the same position. So what can you do with that? A lot of things. One, for example, is to combine it with collagen. And now the question is, will cells still bind and proliferate on collagen? So we actually prepared fibers of collagen and resin and tested the ability of cells to grow. And as you can see, the cells grow extremely well on these fibers along the fibers of the collagen. Now, what about the mechanical properties? So here we have a really pleasant surprise. These fibers, which are made of 95% collagen and only 5% resin, had a 380% increase in toughness and 300% increase in strain at break. So these fibers may be used in the future for artificial tendons or ligaments. So what's for the future? I believe that in the future, we will be able to produce in plants almost everything, not just collagen or keratin or resin. In fact, everything that nature gave us, and then purify this protein in using additive manufacturing technologies to print almost everything, even a heart. Now, of course, this heart is going to be better than one we can get from a donor because we will produce it from better materials. Well, admittedly, this is going to take a long time because heart is quite a complex organ. But sure enough, there are many other organs, such as skin or even liver, that may be used with these materials. And I would like to end my presentation by quoting Pavlov, who said, "If you want a new idea, you should open an old book." And I say that the book was actually written. It was written over billions of years of evolution. And the text is the DNA of life. All we have to do is not reinvent the wheel, but simply read this text and start our progress from there.